Everyone, and welcome. Today we're going to talk about artificial intelligence and machine learning. So let me go ahead and share my screen with you, and let's get started. So what we'll be discussing today includes an introduction to artificial intelligence, or AI. We'll talk about definition and examples, classification of machine learning systems. Then we'll take a closer look at model-based learning, which is what we're most interested in and probably what most of us are most familiar with. And then we'll talk about a variety of different model types that can be used for artificial intelligence. The second part of the talk will focus on some research-based information for you. I'll discuss how I'm using machine learning to do drug design in my research. We'll give you an intro to the project and talk to you about how we're using data and model building in the research for developing alpha helical peptides, how we're implementing many of these ideas that we've talked about in the introduction to AI, iterative refinement, the model that we're using, which is Markov state models, how we interpret the results, and then the summary and conclusions about how that influences our understanding of alpha helical peptides as drugs. So then, let's get into our introduction about artificial intelligence. So, artificial intelligence is all around us. I'm sure you're familiar with many of these companies that I've listed here. Amazon, Google, Uber, YouTube are things that are all household companies that are known to all of us these days. All of these companies integrate artificial intelligence into various things, such as suggestions for you to use and what you should buy next, what you should watch next. Google's AI-powered search engine is an example, advertisements that are making predictions on websites, Amazon prediction for other products to purchase, speech recognition, ride-sharing app, Uber, AI autopilot commercial flights, spam filters, self-driving cars, visual perception, speech, language recognition, decision-making, and translation between languages, YouTube video suggestions, Cortana, and Alexa are a few more of the examples of artificial intelligence that we experience in our everyday lives. So what exactly is artificial intelligence? It's a branch of computer science. It is the study of intelligent agents. That's any device that perceives its environment and takes actions that maximize its chance of successfully achieving its goals. It deals with how computers learn or how to do tasks typically associated with human learning. So there's a variety of different classifications, and it may be useful to have a look at this Venn diagram to understand how the various parts of AI and ML fit together. So basically, artificial intelligence is the all-encompassing idea that we've just been talking about, whereas machine learning is a specific kind of artificial intelligence. And inside of that, we have a deep learning, which many of us think of when we talk about AI and ML. But there are additional kinds of artificial intelligence, and it's important to be aware of these as well. So there is a good old-fashioned AI, which is empirically driven and may not necessarily involve computers at all. It's empirically driven based on what is observed, based on past experience, and the model largely does not change. It's basically, this is the set of rules. Whereas machine learning is going to be based on past and current experiences, and the model has the capability of dynamically changing as new information is observed. So a rules engine, what is this? This is a sophisticated if-then classifier. This is how decisions are made and is often seen in the business world for automation. So we may have some rules that determine how something will be executed and some data objects. These will be the input for a rules engine, which will have those if classifiers. And then the data that's being classified then will pass through those rules, and output will come out on the other side, which classifies the objects into various sets. This is an example. This is nested conditionals. I've taken this from one of my computer science classes. It's a very good example of a classifier. So if we consider some Python code over here, we can see that this is a function which is going to classify shapes based on various inputs. There will be binary questions asked, and those questions will then sort the shapes into one of 4 various geometric shapes. It's of course not all comprehensive because there are other shapes besides these, but the idea is the classifier can classify between cylinders, cones, cubes, and triangular prisms based on asking a series of questions, and one would move through the flowchart in order to determine what the output is. The machine may involve this code over here, which encodes what is being depicted here in this diagram. Expert systems. Information is acquired by experts. A knowledge engineer creates a searchable database, and a user then can search that database using keywords, tags, and general searches. So we have many examples of this. We have WebMD, we have the RCSB PDB, which is a repository of protein data, and we also have PubMed, which indexes many of the articles that we read. The idea is a human expert has some information, perhaps it's a medical doctor having information about a particular kind of a disease. A knowledge engineer then would convert that information into a knowledge base to code that up. Then there would be an inference engine, which then is going to interface between the knowledge base and the user. And then the user, who may not necessarily be an expert, can go in and extract that information simply by using keywords, tags, and searches. So this is an example of expert systems. Knowledge graphs. The idea between a knowledge graph is to depict the representation of how things relate to each other. You may have done this for brainstorming. This is an example of how various nodes are related to each other, and we have various arrows explaining the way in which the information is related to each other. The machine learning approach. So in the machine learning approach, what we're going to do is we're going to have some problem of interest. We're then going to study that problem. We will then train a machine learning algorithm. To do this, we'll have the input of lots of data. We'll define what exactly we mean by lots and how we know if we have enough. That will then provide instances, and then a solution will come out. We'll inspect the solution to see if it has good ideas. Hopefully we'll be able to understand the problem better. If we don't understand the problem better, if the results are ambiguous, perhaps we need to refine the model. And then we make some changes to the model, and then we would study the problem more. Then with that insight and using the hindsight of what we learned from the first time, we would then perhaps be able to pose a better model, and then we would be able to train the algorithm better and iterate through. That's the general approach we would take for doing machine learning. That's a very broad overview, and we'll talk more about the details of what those things actually mean as we move through the talk today. Classification of machine learning systems. There are a variety of different types. We may consider them in terms of supervision, iterative versus batch learning, and instances versus model-based learning. So, is human supervision required is a key question we might ask in order to understand the classification. So we're going to look at annotation of the data. We have supervised, unsupervised, semi-supervised, or reinforcement learning. So let's take a look at an example of supervised learning. In this case, we have a training set. A great example is considering emails. Let's consider this actually as an email filter. So we have a training set. A series of emails arrives at your inbox, and we want to know, is this mystery email a good email or is it spam? So in the training set, we have a series of emails where we've marked them as good emails, which might be indicated to the system, such as we actually make a response to it, or the email is marked as spam, which these ones are being indicated here. So you'll see this is being shown by some tags. And then our job here is to figure out what is the tag on this new instance based on what we have seen before. This is considered supervised learning because there has been a human intervention to indicate what is good male and what is bad male. That's the supervised aspect of the supervised learning. Unsupervised learning, on the other hand, does not involve human intervention. What is being done here is a clustering algorithm. What we're trying to do is we're trying to group people. So there's some feature 1 and feature 2, and these people are then being divided up by some algorithm to separate them out on, it appears to be some sort of a spatial distribution. Perhaps this is town lines or whatever it might be. Unsupervised learning, we can use this for dimensionality reduction. So here we have an example of a number of images, and the job of this algorithm is to be able to identify what these objects are. So we're thinking about facial recognition or image recognition, that kind of a thing. So here we don't necessarily have the object tagged ahead of time, and the algorithm is trying to figure out all these different kinds of possible things that they could be. This is considered to be a dimensionality reduction because the idea is there's a range of what each thing could look like. So for example, if you maybe saw a deer, but you couldn't see the antlers because of the view, perhaps the algorithm would think it was a dog, right? So there'll be some overlap, and so we could say there could be some range of things that we could see. And so what we'll need to do is we'll need to then group together the things that are similar, and those things are going to then represent a dimensionality reduction because we will be grouping them together so there'll be fewer categories. As you can see, the data points are scattered throughout here, and so that means that certain views of certain of these objects could, in some perspectives, seem more similar to something else, and so the idea is to be able to get good at separating out the different objects. Semi-supervised learning means that some things are labeled and some are not. And so image classification, again, is another good example of this. So we might have a robot here whose job is to classify between cats and dogs. So some of the information may be labeled and some of the information may not. And so this is called semi-supervised because, again, it's a question of, is there a tag or available information known about it? And so in this case, since some of the information is, that is why it's called semi-supervised learning. Reinforced learning is another interesting kind of learning. And so this, in this case, we can envision a robot, our agent here. And the robot needs to figure out what it should do. Okay, what's good and what's bad. So what we're going to do is trial and error. First of all, we'll observe the situation. So the robot says, oh, there's a fire, and also observes that there's a bucket and some water. So it could select action using some policy. There's some rules about what it ought to do. So maybe it's equally probable in the beginning to go to the fire and go to the bucket. So the robot chooses an action. Oh, it goes to the fire. Ouch! Realizes that it is very hot and that is a bad idea. So then it will get a penalty. So we get minus 50 points penalty to say, oh, that's a bad idea. I shouldn't do that anymore. And so therefore learning from experience not to do that again. And then next time it will choose to do something else. And so then what it will do is it will iterate through doing various policies. So it will make a different choice. Maybe next time it will choose to explore the faucet over here. And who knows, maybe eventually the robot will realize that the water can quench the fire and make things even better. And so this is the idea behind reinforced learning, is to try to do something better than what you did before by the penalty and reward system. So another way we can classify systems is, can the machine learn incrementally on the fly? So if, yes, it is learning on the fly, that's called online learning. And we can also have batch learning when we can just give it one batch and then it will learn from that only. So in online learning, let's try to understand what that might be like. So here we have some data and we're going to train a machine learning algorithm. Then we evaluate the solution. If it's great, we'll launch and then we can run and learn some more, and then we get new data on the fly. And so that data then can be incorporated into the analysis, and it could be used to iterate if needed. So that's considered online because as we're going along, we're generating new data that will give us additional information that could be used to train and improve the model. Batch learning, in contrast, does not allow for new data on the fly. That means that there's a single data set and that's the only data set that's available. No new data is going to be provided to the model, so it will be just only as good as the data set that's available. Maybe this could be representative of, maybe you could only take one sample. Maybe it's, maybe it's some particular event, maybe it's, some geological event, and then, you're studying a volcano and then the volcano erupts, so you cannot get any more data about what the state was like prior to eruption, after the eruption, something like this. Online learning for huge data sets. So one of the things that is very interesting to do is we may have much more data than we can handle at one given time. Perhaps there are limitations to our machine, limitations of the processing, limitations to resources available. So what can be done is we can chop the data into some smaller pieces, and we may be able to train an online ML algorithm to evaluate the solution, analyze the errors, and then study the problem, and then give it a different chunk of data. So we might be able to iterate through or iterate into various pieces of data, and then when the solution is satisfactory, then we would be able to launch and use that model to make predictions about whatever questions we're trying to answer. Another way we classify machine learning systems is, does the machine compare new data to known data or detect patterns and build a predictive model? So we have the distinction between instance-based and model-based. So instance-based learning is like this. We have some data, some triangle, and some squares. And what we now have is some new instance, and we want to ask the question, is it a triangle or is it a square? So machine learning is going to look at specific cases and then check new ones using some kind of a similarity measure. Model-based learning, on the other hand, is going to be based on some kind of a model. So instead of comparing to known data, what it's going to do is it's going to surmise that this dotted line is going to divide the different types of data. And so maybe this model has a specific equation to map this boundary. Maybe it was obtained by some particular method. And then what we would do is we would want to understand whether or not this new instance should be considered a triangle or a square. So model-based learning is primarily what we're interested in, and oftentimes what we think of when we're thinking about machine learning. So let us take a closer look at model-based learning. So what is the model? The model is a rule to be used to generate predictions. We make models all the time. The model can be as simple or as complex as you'd like. Some models are better than others, so we could say we're going to have a model that's a random number from 1 to 5 will be the model. This is how we're going to predict the temperature for tomorrow, right? Do you think this is a very good model? Probably not, but it is in fact a valid model. So some models are going to be better than others. We can often do this by fitting data to obtain an equation. I put up some equations that you may have recalled from maybe your pre-calculus class. Data can be fit by many different kinds of mathematical functions. And then what we can do is use those functions and we can assess the quality of fit, which will depend on the data that is available. So to consider a model-based learning, we could look at a life satisfaction example. So in this study, which is explained in this link here, if you want to learn more about it, there was a variety of countries, and some data was collected on the people in these various countries. So this is the GDP per capita, which is apparently some measure of wealth. And over here we have life satisfaction. So we might say, does money make people happier? This could be our question of interest. And so we might apply some model-based learning to find out whether people are happier when they have more money. So what we can do is we can do a binary classification or support vector machines. We can ask in this one, is X a circle or a square? So what we want to do is basically draw some kind of a line here. So these are some axes, and we want to be able to draw a line that will separate out the circles from the squares. So what we're going to do is decide if some input is one of two options, whether the X is a circle or a square. Example of supervised learning, that is classification available for training data. Then what we're going to do is draw a line between the data that best separates out the categories. So we see here a line has been drawn. We can say, okay, yeah, reasonably good. More complex variants may use other equations besides just a straight line. So here's an example of another equation. Maybe this is one of those other functional forms we talked about. Maybe it's a linear combination of lines, whatever the model might be. And then we would plot new data and look to see how it matches. And also, once we get convinced that it's able to separate between them, then we may be able to use this to make predictions about unknowns. Okay, so then getting back to our model-based learning with life satisfaction, we could study some data to get some hypothesis. So we're thinking here and say, oh, maybe it's linear. Maybe people's life satisfaction is directly proportional to the amount of money that they have. So let's make a graph. Here's life satisfaction on one axis and the GDP per capita on the x-axis, and then we plot the graph. So now for model-based learning, what we're going to do is we're going to try to write an equation. So this is our actual model, and we're going to iteratively find it. So there are going to be some fit parameters. This theta 0 is basically the y-intercept. And then we're going to have theta 1, which is going to be multiplied by the GDP per capita. And so then we can assess how well it fits the data, and then we can tweak the model, which is going to amount to changing the parameters. So then we can turn this into an optimization problem where we attempt to improve the fit of the line to the data. So we're saying this is our equation. How can we fit that line better? Well, we can change the variables that are in the line, right, which is the theta 0 and theta 1. And then we can see if it fits the data better. And if it does, we can accept the move. And if not, we could throw that out. Next thing we need to understand is the fitness function. So typically, what we're trying to do is we're trying to answer the question, how well does the data fit the model? So if we have some data here, right? And here are some data points. And so what we want to know is, well, how good is this model for fitting the data? So what we would do is we would pose a variety of different fit curves. These blue lines here, you could imagine moving this around. And then we would need to calculate how well it fits. So a typical thing to do is to assume that all the error is in the y direction. And so what we do is we take the difference between the current x values times the difference in the y values over the x minus x bar, the average value squared. And so this will give us, well, this is basically a root mean, a linear least squares example. Those of you familiar with root mean square deviation, it is very similar to that formula. And so what it will tell us is how well this fits. And so the distance will be at a minimum when the best fit has been found. So how do we know when the refinement is complete? So in fact, actually, there's no way to say it's quote done. The ability of a model to predict a particular data set can always be improved by adding more parameters at the expense of becoming overly complex. Imagine drawing a line to separate two categories. How could we make it more and more perfect? Well, instead of drawing a straight line, we could say, well, I'll draw a squiggle, right, freehand, and then you can draw around the data points that are not in the correct orientation. But the problem is that's going to be subject to overfitting because you're looking at an example that's too specific. So generally what we're looking for is the minimum distance between the predicted model and the value of the actual data. So then to summarize how model building works, we first collect data, then we generate a hypothesis, then we'll select a model, and then we'll iteratively test and refine our ideas. And that may involve selecting a different model, tweaking parameters, or minimizing error using the fitness function. We iterate the process as many times as needed, and we attempt to get a model that best fits the data that we're interested in modeling. So next we need to talk about the training set, the validation set, and a test set. So when we make a model, the set that is used to actually generate that model, especially if it's going to be based on some data that it's observing, that training set is going to have a lot of influence on what the model is capable of doing. Therefore, what we need to do is we need to make sure we haven't overtrained, that is made it too specific to find only particular examples. What we would do then is we would use a validation set in order to find out what the answer is. The validation set is not known to the machine what the classification is, but it is known to the scientist. So if the validation set is correctly classified, it builds our confidence that the model is doing well and is not overfit. Finally, once we are sure that the model is working well, we can then use it for predictive work, where we can use a test set where in fact the classification is neither known to the machine nor to the scientist. By having the good validation score, then it increases the confidence and helps us believe the data that we're looking at. There are a number of challenges to machine learning. Insufficient quantity of training data, non-representative training data, so you could have a lot of data, but it could be taken only from a very small subset. Poor quality data, overfitting the data, or underfitting the data. Additionally, we've talked about some specific model types, but there are more model types beyond that. So the idea is that the model types could be interchangeable. We think about our process here for doing artificial intelligence and machine learning, and imagine a film strip that could pass through and stop on a different frame and could plug in a different model for that, select the model part in here, right? So we could collect data, generate a hypothesis, and then select a model and iteratively refine it. So more than one model may be appropriate for a particular problem. Some may be better suited than others. Decision trees are another kind of model. This is an example of supervised learning, an example of more complex classifier than support vectors. And so we'll have more decision points in here. So instead of being just binary, notice that we have multiple points in which decisions could be made. This allows greater flexibility, and this does not restrict the specific number of answer categories. That means there's no restriction on the number of classes. So here is an example, and at various points you're going to classify by a yes or no question or by a multiple choice question, but iterate through the branches until a leap is countered. That is the end. This is similar to what we were talking about in classifying the shapes. Ensemble learning and random forests. So there could be a couple of different classifiers that are all constructed in a slightly different way. maybe we don't know exactly what the architecture is. Maybe we're not completely sure. So what we might do is we might try out a bunch of them. We might make multiple copies, and we might try out different architectures, different ways of asking the questions that would also turn out what the answer will be. So the trees are the different models, and we're going to begin with a randomized structure, which each are iteratively refined to fit the data, to figure out where the branch points are and so forth. We then have multiple architectures for the same data, and that could introduce some variation into the trees. So the trees is referring to the various models that are being used here. They're going to be constructed for multiple decision trees, each with their own architecture. This allows for greater variation, and it does not restrict the problem to a single tree architecture. So then we can do many different trees, and then we can then take the consensus view, and that is what will determine the overall answer. Markov Chains is another example. This is when we have models has a known number of states. So you might have a child who likes to sleep, run, and eat ice cream, and then we might ask what is the child doing now, and what is the probability that they're doing each of these things, and that after they do a particular one that they transition to doing another activity. So after they eat ice cream, we have a very high probability that they're going to start running around. And so this gives us an idea of how we might set this up. Markov chains answer the question, what will happen next, and quantitates this with transition probabilities. This is an example of supervised learning. So we're going to learn the patterns of the transitions based on the observations, based on what happens next. The next action will depend on probabilities in the current state. This is very useful for analyzing data that has sequence. Hidden Markov models are a variation of Markov models in which some states are not known. Another thing that's different about hidden Markov models is that the states have probabilities within them. So for the MSM states, each of the states had an associated thing with it: eating ice cream, sleeping. There wasn't the possibility to do something else. Whereas in the hidden Markov model, each of the states has a probability to be anything of many possibilities. Is just that the different states differ because the probabilities are different in those different states. So for example, we may be interested in studying the sequence of a DNA that a protein binds to, which is being done here. And then we might consider what probability do we have in each position of the DNA for a particular DNA sequence to be there. So we can use this to classify sequences of events or data, such as multiple sequence alignments. This is going to represent noisy patterns that are found probabilistically. Each state has its own probabilities associated with it, which pertains to the probability of a particular position in the alignment. This might make sense because proteins, for example, might be able to select a particular position for a certain residue to be there because of the hydrogen bond pattern that they have. Whereas in a Markov state model, that's really not a consideration that we can take into account. The alignment can account for insertions in the sequences or deletions. This comes from the idea of biology, where we would have in the genetic code some insertions or some deletions of particular DNA bases. The insertions are these I's down here, and so that means that an extra thing may show up, and the D stands for deletion. Both the alignment and the probabilities are learned as part of the training. The model has both emission probabilities and transition probabilities. The emission probabilities means the probability, for example, to have a certain DNA base in a particular state. So these would be the emission probabilities. And the transition probabilities are the probabilities associated with traversing the various arrows. It's going to tell the optimal path through the model, as well as the likelihood it matches pattern provided in the training set. We also have neural nets for deep learning. A neural network is going to combine multiple nonlinear processing layers using simple elements that are going to operate in parallel. This is based on a model from the biological nervous system. It contains an input layer, several hidden layers, and an output layer. The layers are going to be interconnected via nodes or neurons, and each hidden layer using the output of the previous layer as input. What it means by hidden is that we may not actually get the information out. We may not know why the probability is that, just that it proceeds through these various states. And then what we're really interested in is just the input and the output. How does a deep neural network learn? So let's suppose we have a set of images where each image contains one of 4 different categories. So here's our images. And we want the deep learning network to automatically recognize the object that is in the picture. We can label the images in order to have a training for the network. Then we can use the training data, and the network can then start to understand the object's specific features and associate them with the corresponding category. So we don't necessarily know what features would be found in the beginning, but what you can see here is for Lily, it's been divided up into several components. And so what we see here is that it should have this general overall shape. And that might be something that's a feature of it being classified as a lily. Notice that the network learns directly from the data. There is no influence over what features are being learned. So various features could be learned based on the different inputs that are provided. So deep learning is a subtype of machine learning. With machine learning, you can manually extract the relevant features of an image, but with deep learning, you can feed raw images directly into a deep neural network that learns the features automatically. Deep learning also often requires hundreds or thousands and millions of images for the best results. It's also computationally intensive and requires high performance computing use. And we talked about working on the cluster in some of our previous lessons. Machine learning and drug design. So now that we've had the opportunity to talk about machine learning and what it is, today I will take the opportunity to tell you about a project that I've been working on. In this project, what we're trying to do is we're trying to understand alpha helical peptides, which are a possible type of drugs. So in order to understand this, let us consider the problem that we're interested in solving here. So we're interested in developing a new class of drugs called allosteric effectors. What we want these to do is modulate the activity of an enzyme while maintaining the use of the active site. This is our typical cartoon that we show in biology classes to understand what allosteric signaling is. And so here is an allosteric protein that has these 2 binding sites. So we want this to bind to its green ligand, but you see that it's closed here, so the ligand's not able to bind. What happens here is that the triangular shaped one binds to this, which causes a conformational change in a distant part of the protein, and then it's able to bind to its native ligand. So the allosteric effect then is this purple drug binding here, causing this to have the conformational change. It's now in a binding complement state. Upregulation and downregulation would be possible, and activation and or repression. We could get a rheostat effect, but not just binding on and off, so that if we were to bind in the active site, we may have only one option, either it's blocked or it's not, whereas this may be able to adjust it in a more more of a range, so that you could have more effects. You could have higher selectivity for a specific number of protein family. These are generally considered to be safe for drugs because they may not be so strong that everything is always just one way. And drug targets that are not currently druggable may be druggable by allosteric effectors. Some of the current challenges to allosteric drug design are detecting where the allosteric points of control are located. So we don't necessarily already know where the protein will be allosteric. We need to understand how the allosteric signal is propagated in the system so that an appropriate effector can be chosen. We want to know whether an activated or repressed conformational state is pre-existing in the ensemble of the unbound structures. So proteins are moving all of the time, and so we want to ask the question, is the shape that is ready to bind present in that ensemble, that various mix of structures is available or not? And allosteric effectors are often found serendipitously. We'll be talking about the P53 system. So P53 is the guardian of the genome. It's also known as a tumor suppressor protein. Its role in the cell is to select between cell cycle regulation to arrest the cell from dividing, or to cause the cell to commit apoptosis. So imagine that a certain cell underwent some DNA damage, and so the cell is now no longer functioning properly. What would we want P53 to do? Well, it should go and it should either stop the cell cycle regulation and cause the DNA to get repaired by recruiting the DNA repair enzymes, or the other thing that might happen is we may want it to simply just cause the cell to die, so that we'll just start with a clean new cell that doesn't have those problems. If bad cells do not get stopped, then we can see that we could pass on bad genetics, and so then we might have lots of cells that are unchecked in their growth and are not differentiating into anything particular, and that is a tumor. So this is an example of the P53 structure. It has an N-terminal and a C-terminal region, which are the two sides of proteins, and then we have these major features, some loops that are reaching down into the DNA and interacting with the DNA in order to bind to it in a specific manner. It also has a zinc associated with it. We'll also look at the MDM2, which is a regulator of the P53 protein. We do not have the total structure available, but what we do have is a part of this structure available where we can see a piece of the P53 is being bound by the MDM2 regulator. What is a stapled alpha helical peptide? These are the kinds of drugs that we're trying to design. So typically when we have a normal peptide, which is a piece of a protein, one of the conformations that it could have is this alpha helical shape. So a regular alpha helix would not have this link between the parts. This is called a stapled alpha helical peptide because the side chains that are coming off of the peptide can form a covalent bond and therefore will lock this alpha helical peptide into that shape because it's linked between the two sides. This can be achieved by making some artificial amino acids, this MK8 and OEH, which are put in here, and they have very long side chains that reach off. So this purple part is where the tips have joined, and this blue part is representing this long body here, which is going to stretch down and across over that, that divide there. And then the chemistry of these is allowing the ends to become linked so that it locks the peptide in that conformation. It's called a stapled alpha helical peptide. This is an example. We can do MDM2 inhibition using peptidomimetics. The idea behind this is that we may have an MDM2 which may be overregulating P53. The problem with this is that P53 being sequestered by MDM2 is not free to go and do its job. So if MDM2 is unable to release P53, that could be one kind of way you could get cancer. The idea behind this is if we could put in a sort of like a fake peptide so that MDM2 would bind up the fake peptide preferentially over regular P53, would be able to release P53, which would then be able to go and do its job to either cause apoptosis or DNA repair. These are some structures of that actual peptide. So alpha helical peptides can successfully bind and compete with MDM2. This is an example of something that's going and binding in the spot where P53 is normally supposed to be. So this is the MDM2 regulator of P53, and it would like to go around and bind P53 right in this cleft. But this here is the peptide that's been designed to match to it, and that's binding here instead, which means it cannot bind to P53. And so if a person has the problem where MDM2 would not let P53 go free to do its job, it may prefer to bind to this peptide and thereby release the P53 and be bound up with this peptide instead. That could release P53, and then the tumor suppressor protein could do its job. A first attempt at understanding stapled alpha helix efficacy as a competitive inhibitor with MDM2 for P53, we might try to do this with decision trees. So here is some experimentally available data about the sequence of a variety of different peptides. These peptides have some variation in terms of their sequence. Associated with these is a charge. So if you're familiar with the pH scale, you can see that pH can cause various charges to occur because of the concentration of hydrogens available. So the point is that they can have varying charges. This is the amount of alpha helicity, that is how much they look like that alpha helix twist. Kd is the binding constant, so this is a number that's readily measurable. Then the next question we want to ask is it cell permeable? So that's an important question, because if a drug cannot get inside of a cell, it's not going to be a very good medicine no matter how well it binds. So all these peptides here are not able to get into the cell membrane. Notice that it's pretty close to matching up with the charge, so we realize that having a negative charge is probably causing an issue with it to go across the cell membrane. And then the next thing we want to ask is, did it work? So if the peptide goes across and the cell died, the presumption is that it successfully bound to MDM2, released P53, and caused the cell to die. Out of this whole set, only one peptide worked, and that was the SAH-P53-8. This is a diagram of a peptide going across the membrane, and this highlights some of the problems that we still have. Much of the work is going to be trial and error, so we don't actually know the rules about how this is going to happen. Only one of the peptide works, and we can understand molecular principles only if we understand how they actually operate. What we might do is wonder how network theory could be used to address the problem, and then maybe try to construct some networks. Let's have a hypothesis that peptides having a higher alpha helical content permeate the cell membrane. So here we're going to take a look at the role of charge. So in order to permeate the membrane, the peptide should not be negatively charged. So these are the peptides that have a -2 charge, and they also do not permeate. So you may make the assumption that peptides should not be negatively charged. This is just a depiction of the fatty acids in the lipid membrane. So here what we're going to do is we're going to have our sample number, so just the peptides that we studied versus the binding affinity, and then we could say, well, do we have any trends here? We see that we have SAH3 and, all right, so SAH8 is the one that we know actually worked. This is our winner peptide. That one is permeable. SAH3 we see is not permeable, as are all of the red dots. So these ones over here are the ones that are more interesting. We may also make a hypothesis about the alpha helical content of the stapled alpha helical peptides. So we've already eliminated some, and so what we're then going to do is we're then going to continue on with our data search. So we're going to look at the percent alpha helicity, and we're going to compare that to the sample number. Again, the red ones are already eliminated, so SAH8 has a very high alpha helical content. We see that because it's way above the others. So that hints to us that maybe that's a special feature. We could use a decision tree to try to understand something about this. We say, does the peptide penetrate the cell membrane, yes or no? And if it does it bind with sufficient affinity to be used as a drug? And if we say no, then the problem is that it wouldn't really work as a drug. But if we say yes, we just say, is the alpha helical content greater than 50%, which you might have learned from empirical observation. If we get yes, then we would see we could select for SAH8. So this is an example of a decision tree that could be written using empirical observations and those hypotheses that we made by just looking at the data. MD-MSMs of alpha helical peptides. The aims of this project were to characterize the conformations of the alpha helical peptides. You could answer questions: do peptides sample other energetically stable conformations? How do peptides transition between available conformations? And is being able to form the conformation of this H8 sufficient for function, or does residence time matter? We can also gain useful insight that we translated for other drug design projects. We'd like to be able to gain insight into the principles that play rather than just rely on luck or trial and error. And this is going to use competitive inhibitors for other systems. This is how the stapled alpha helices were made. These were generated from the Verdine peptides, that set that I showed you before. This is going to be a composite of all the mutations that were made, and is showing where all the possible mutations could occur. Then we'll run a molecular dynamics simulation. MD simulation is essentially just based on using F equals ma many times, and the positions of the molecule are going to move in time, subject to a force field. Here we're showing what the force field is. The protein mutations will occur. You may need to model in DNA sequence changes. You may need to model in ligand, drug, cofactor, or whatever else may be in your system. And we would put the hydrogens on the system and introduce ions for electroneutrality, and then add a box of water so that the molecule will be solvated, and that's what's being shown here. The next thing we do is measure the root mean square deviation. This is very much similar to what we were talking about earlier about fitting a model and how well it's doing. Basically, we're using almost exactly the same equation to measure how different a current snapshot in time of our MD simulation is as compared to the initial starting structure. We've done this for all the different peptides in our set. So let's consider this toy model in order to understand how this is going to work. So what I've done is I've reduced this down into representatives of the peptides that are going to illustrate the major things that are going to happen with the peptides. So let's consider that this arrow represents the MD simulation time, and so we have a variety of snapshots. Here we're showing 10 snapshots. Of course, there are many more snapshots than that. But in this, what we're going to have is a variety of snapshots that are going to have different shapes, which is suggesting that they have different conformations. This says that there are 4 substates are present in 4 different MDs. So here are the 8 MDs, and the 4 substates are the 4 different shapes that are represented. Some are going to be found in more than one MD. Our goal then is to group the substates into bins. So we want to put all the circles together. So all the times the protein was in the circle conformation, all the times it was diamond, triangle, and square, respectively. One of the problems we may have is not knowing the correct number of bins. So if we do not have enough bins, the problem we would have is that the difference between the shapes in the same bin is too large. So then we would need to have more bins. If we had too many bins, we would have similar substates that are being split into different bins. So the distance between these snapshots and these snapshots is very similar, and so they should be recombined. This suggests some iterative model refinement that can be done. So what we would do is we would guess the number of bins. We would then sort the snapshots into the bins. Then we would assess the similarity of substates within the bins. That's basically going to be a fitness measure. Here we're iterating over this as many times as needed. We know that the number of bins is optimized when the number of bins is large enough to accommodate each of the different substates, but the number of bins is small enough so that each bin has a distinct substate. This is an example of such data where we're having a different number of bins in which we're clustering, and we're showing the distance between these snapshots and their respective centroids in the cluster. This over here shows you an example of some of the different structures that would arise in some of the different centroids. What does it mean if MDs in the same bin overlap? So that's a very interesting question, and this says a lot about how Allosteric might be working. So for example, here we see that we have all these green circles. It says that SAH8 always does the same thing. It only has one conformation. SAH4 has mostly those same conformations, but introduces another kind of conformation too. So when we have an overlap, what that means is it means that the conformation is shared between the different simulations. It didn't matter that these snapshots came from a different MD. This is just the conformation that it's able to access. The sequences of SH, SH4, and SH1 all have the ability to adopt the circle conformation, whereas SH4 and SH1 have the ability to be in the diamond conformation, but it's not accessed by SAH8. So then what we might want to do is sort the data in each of the bins by MD once we've figured out how many we have. And so then what we can do is we can directly compare between the counts to say something about how probable something is. So for example, SAH8 only does a single conformation. And so here what we have is that although it's accessible by others, SAH8 has the highest ability to form the circles. It means that it has the same conformation sometimes in these other simulations as well. The snapshot distributions then are going to be an important way to analyze the data. This is the distribution of the snapshots between the clusters from our trajectories. And so what we see here is that state 1 is going to have our SAH8, the stapled alpha helix number 8, the one that was the winner from our studies about going into cells and actually causing cell death. There are some other peptides that do similar things, but many do different things besides that. So SAH3, for example, is almost also 100%, but it's in the wrong conformation. So that's why that one is not a particularly good one. This blue one here is similar to SAH8, but this one, for example, didn't do that all of the time. Being able to adopt cluster 1 conformation is not sufficient because all of these other conformations do access cluster 1, so that means that's not the property that's being checked here. Rigidity is also not the operative factor because SAH3 adopts a single conformation. So we may say, well, the reason might be because it only has one conformation, so does SAH3, but it's the wrong one. And so that's why it's not permeating the membrane. So we can heuristically interpret the data to understand what might be a good feature. We can also consider the transitions. So the transitions are going to be the probability to move between those different substates. SAH8 is going to be almost exclusively alpha helical peptide and also transitions out very few times. So again, SAH8, the winner, got to be so good, as we're observing from here, by not really moving into any other conformation because we don't see any bars in the other ones that match this teal color. And also the transition numbers are very low compared to staying in this state here. So 480 times it will remain in the substate 1 circle, and very few times it will even transition elsewhere. So small that we don't even see the bars on the scale. Okay, let's consider a different model. So in this case, this SAH4 was in the circle conformation some of the time, but also spent a lot of time in another conformation. And so we see the evidence of that as it transitions over into other conformations. Being sometimes alpha helical, but mostly in the wrong substate, this means that flexibility does not help. So here's our green bar mostly being in substate 2, but sometimes being in substate 1. So that isn't a winner, so it works only some of the time. Here's another example, which is SAH1. Being in lots of substates, including alpha helical, is what characterizes this one. So could doing lots of different things be great and helpful for binding? So no, because this one transitions between many, and we know that that was not the winner. So multiple substates with deep wells are not the way to go. Yellow, this one here, SAH3, is almost always non-alpha helical. So again, we could look at that idea of being in just one substate. Is that the thing that's operative here? And the answer is no, because this one is almost always in substate 4, but that's not the correct shape. So therefore, that's not the reason why. And so it's not because it has rigidity. It has to do with being rigid in the correct shape. So then we can come back and we can take a look at the Verdine data one more time and consider that in light of what's going on here. So we see that SAH8 is the only peptide that was a winner here by permeating cell and causing cell death. We see that we may have some rules about becoming cell permeable, particularly pertaining to the pH. And so here our data is telling us information from the molecular dynamic simulations as to predicting those peptides. So then we can go back and review the machine learning process. We have alpha helical peptides in mind. So we first collect some data. So we had the RMSD data and we had literature information. We generated a hypothesis about how the alpha helicity might be important for permeating the membrane. We selected a model. So we did Markov state models, and then we iteratively test and refine. So we select a different model, tweak parameters, minimize error using the fitness functions. So there's a variety of ways that we could improve this by selecting different numbers of bins and so forth. So then the stapled alpha helices summary and conclusion. The stapled alpha helical peptides are a variety of conformation evidenced by the various substates that we saw. The first conformation corresponds to SAH8, the peptide that successfully inhibited the MDM2 in vivo, as was known before we started. Peptides passing across the membrane are all adopted conformation 1, SAH8, which is the only one that both penetrated and was active, was almost exclusively in conformation 1, that alpha helical shape. So in order to be active, having reduced configuration entropy, that is, being in only a single conformation, may be what is most helpful. We've also done a variety of other contributions to allosteric drug design and machine learning that we didn't have time to talk about. We're using graphs and network theory to understand pathways in the US, which is one of the largest proteins known. We also did pathway analysis in proteins, where we used decision trees to understand the paths, physical paths between residues that may be used to transmit allosteries. We asked the question about their frequency and how readily they're able to be formed. We use graphs and network theory in MD sectors to analyze what the cohesiveness of an allosteric network. And finally, we're using MD Markov state models to understand the various substates and transitions between them, as we talked about in the alpha helical peptide. This is being shown for our model system PBZ. So these are our acknowledgements. Thank you very much to our colleagues and coworkers, and to the many sources of funding that we have. Right, and I would also suggest that you have a look at O'Reilly.com. This is a book chapter that a lot of the information in this was based on, so you may wish to read that for yourself, and it's available online for free. All right, so thank you very much. I hope you have enjoyed the talk about machine learning and have a good overview of how machine learning works. In the upcoming lectures, we're going to explore a variety of other topics. We'll be talking about Markov state models, hidden Markov models, and neural nets in more detail. So I look forward to seeing you in some upcoming workshops. And till then, bye-bye.